Why did we use strawberry? Strawberries are often used in DNA extraction experiments since it contains 8 pairs of chromosomes, which will give a fast result compared to bananas, which has only 3 pairs of chromosomes. Most of all, strawberries are soft and are easy to crush, but since there are no fresh strawberries in our province, we used frozen strawberries that we bought online in our area. First, we need to mash or crush our strawberry in a Ziploc bag for about a minute. We transferred location because our first setup was on top of a glass table and it might break when we crush or mash our strawberries. We actually used a blender here to finally crush the frozen strawberries because we had a hard time crushing it manually because it is frozen. Now, because the strawberries were sticking to the sides and the blender was not working, we decided to place the 1/2 cup of water in it. We then added it with 2 drops of dishwashing liquid. Then we added it with 1 tablespoon of salt. The salt will help the histones dissociate from the DNA found in the nucleus of the cell. We carefully mashed our mixture together to avoid foaming. We then filtered our mixture using the paper towel. The soap helps us to dissolve the cell membrane, which is a lipid bilayer. Sodium chloride helps to remove proteins that are bound to the DNA. It also helps to keep the proteins dissolved in the aqueous layer so they don't precipitate in the alcohol along with the DNA. We place the filtrate in a clear glass. Then we superimposed an equal volume of ice-cold rubbing alcohol. We then wait for 5 minutes to see the result. Why do we have to use ice-cold rubbing alcohol anyway? It is important to use cold alcohol because it allows a larger amount of DNA to be extracted. If the alcohol is too warm, it may cause the DNA to denature or break down. The colder the alcohol is, the quicker the DNA will be extracted.